But I wanted to create a couple of videos to help you go through the various different steps of doing assignment number 1. In a prior video, I showed you how to do the initial blast search to find that Fo13 has a human analog that's glycerol 3-phosphate phosphatase. And we're going to go ahead and build everything based on that. But this alignment you saw, and this is the results you saw. These are the results you saw based on that blast search. You see there's a lot of relationships here. You see a lot of amino acids are in the same. If the letters are here, that means they're the same in both sequences. And a blast search just finds big relationships. It might not find smaller relationships. So what we wind up doing is we wind up taking those sequences we get from there, and then we go ahead and go to another software, usually Clustal Omega. Nice thing about Clustal Omega is if you want to align 3, 5, 10, 256 sequences, you'll be able to do that in that software. But the first step in doing that, what I always like doing, is creating a file that has all those sequences in it. So I'll bring in a little window here that from a text edit program. I'm on a Macintosh right now, so you'll keep in mind that I'll be using text edit, and if you're on a PC, you probably use WordPad, works very well. The key is all these files need to be kept as text files, or in the jargon of Macintosh, a Unicode 8 file. So here we have here, and what I have here is there's the Fo13 sequence. I'll copy that. Command or Control C. Command or Control V, paste it in. Hit a couple of carriage returns. And then I'll go to the phosphoglycerol phosphatase sequence. Copy that. Paste that in. And what I'll do is I'll go to the view here and zoom in. I don't like how that does that, but I want you to be able to see the characteristics here. For this file, looking at it, you want to have a greater than sign and something like Fo13, so you know that's what the sequence is. And then human PGP here. And you see here's the sequence. So we'll go ahead and file. We'll save that. I have a tendency to name things based on the next step in the process. So I'll call it Clustal Omega input file. So what this file has is it has both Fo13 and PGP sequence in it. I'll go ahead and close that off. Here, I gave you the website and the assignment. I already had it loaded up to make it a little bit quicker. Here, I'll go ahead and choose the file, Clustal Omega input file. Click open. The next step here, and this is an important step, this has Clustal W with character counts. If you were only going to be doing just Clustal Omega and not do anything else, this is not a bad alternative. But the better one is to use just Clustal W because it doesn't have extraneous things in there to confuse the next step that we're going to use. And go down here, more options. This works out pretty well as it is. If you're doing a lot of detailed work and want to change a few parameters, you can do that here, but don't worry about that now. So go ahead and click Submit. Whenever you hit Submit, it'll tell you it's queued up. It'll be running shortly, depending on the time of the day and how many other people are working and all those kinds of things. You'll see varying different times for this to be done. Here it's done. Good. So here I'll do Command plus, make it a little bit bigger so you can see a little bit easier. You see there's sequences that are lined up. FO13 being on top, PGP being in the, sort of in the middle. And wherever there's an asterisk, that means the amino acids are the same. Where there's a period, they're somewhat related. And if there's a colon, they're more related. They're more similar structurally by being an amino acid group. So what I'll do is here, I always go and click this download alignment file. And what I do is I'll copy all of that, Command or Control C. I'll paste that into a file. I'll zoom in a little bit so you can see what it looks like. So it has Clustal, and these are all important little features. It has Clustal multiple sequence alignment. It has the asterisks and colons and all that sort of thing, all this sort of stuff. So we'll go ahead and file, then we'll hit save, and I call that Clustal output file. So while I'm doing this, I create one folder, and every search has all the different files in it, so that if something didn't work, or I see something odd, or I find that maybe I had the wrong sequence, I can go through and make some minor changes and figure it out. Just go ahead and hit save. But the key here, the first step along this process, is to go ahead and generate this Clustal file. But if you look at it, even though it shows these relationships, it's kind of hard to see the relationships and see how much is closely related and not. I mean, if you looked at these a lot, maybe you would see that, detect that, but I think it's always kind of nice to take it one step further. And there's a program out there called Boxshade. And Boxshade takes these relationships and creates boxes and color codes the sequences and the alignments for you. So what we'll do here is we'll choose the file. And that's, remember we did the Clustal omega output file. And as input format, it's going to be in the Clustal file format, even though our extension is probably going to be txt. Go ahead and tell it that's the format the file is going to be in. We want to go ahead and write the file in rich text format because that allows us to do some editing. If we want to change colors or anything like that, we can do that. Always like adding a ruler to the alignment, so along the top of the alignment will be sort of relative amino acid positions. And the sequence position at the end of each sequence, you'll know it goes from residue what to residue what, which is kind of nice information if you're particularly trying to link it back to making a mutation. I don't click either of these boxes. No consensus line. It's not a DNA sequence. That's fine. The fraction of sequences must agree for consensus. That's fine. And output width is 60. So let's go ahead and click Start Shading. That goes really quickly. And then we go and click here, Download. I call this Boxshade output. Save that. And what we'll do is I will go ahead and use Word on my Mac. And we have Boxshade output, RTF. Go ahead and open it up. And here we go. This is the desired product. You can go ahead and see here, you can see areas that are obviously very closely related because there's a lot of amino acid identity and things like that. And the gray areas are pretty similar. And this is kind of a nice thing. And if you want to, you'll be able to, like for example, for some reason you want to change the color of those to change that to red, you'd be able to do that. I'll take that away. And so this is part 2. This is the output of the first part of the assignment of the Clustal omega alignment between these two proteins. We'll be using this information to help us figure out the mutations we're going to make and get ready for assignment number 2. There's also a video on using a software to go ahead and do DNA and protein sequence alignment, and I have that in a subsequent video. If you need any help or anything, let me know. Give me, send me an email, drop by my office, or you can ask questions in the lab. We'll see you later. Bye.